So that's been nice and short, which is very good because it leaves us more time for today's meeting, which is the annual debate 2018. And the topic, as I'm sure you're all fully aware, is synthetic biology. And I'd just like to say that this is one of those fantastic joint collaborative ventures, and it's in association with the London Evolutionary Research Network, or LERN, which apparently was established in 2002, and is really thriving. So it's, I think, great that the Society has such a good interaction with LERN. Just going to say a few words about synthetic biology, which are not my own words, but have been written for me. And then I'm going to hand over to the moderator for this evening's meeting. So recent technological advances are allowing scientists to redesign organisms or even build new life forms from scratch. This new developing area of research steps beyond traditional genetic modification, or GM, to combine science and engineering in what is known as synthetic biology. Technologies such as CRISPR-Cas are already allowing us to apply GM to a wide range of fields, from pest control to human genome editing. But these potential practical applications are huge, such as bioengineered micro-machines, microorganisms that can destroy cancer cells, detect toxic chemicals, or produce drugs that are otherwise incredibly difficult to obtain from nature. And then there are a series of 3 questions. Is society ready for this new discipline? Where should the limits be set? And what are the benefits and the risks of these by design organisms? So I'm sure that each of those 3 questions will be explored this evening. And now I would like to hand over to our moderator. Where is she? There she is, Katrina Khan, who's a PhD student at the Institute of Healthy Aging at UCL. And she's part of the organizing committee of LERN, who have essentially put tonight's meeting together. So thank you very much, Karina, and looking forward to your moderation.
Yes, and thank you for the introduction. So if our speakers would like to get ready, I quickly give you the order in which they're going to present. So it'll be a short 10 to 15-minute presentation, followed by a few questions from you and some discussions. So we could start with that. So you're going to first hear from Professor Robert Edwards, followed by Dr. Louise Horsfall, followed by Dr. Pablo Carbonell, and finally Professor Jim Dunwell.
Okay, thank you, moderator, and thank you for the invitation to come along today. I'm Professor Rob Edwards. I'm the head of agriculture at Newcastle University. And to me, synthetic biology in the terms of agriculture is the introduction of engineered, designed traits into crops and animals. So that's very different from what we do at the moment, which tends to be one gene at a time. And maybe rather than just addressing the specific questions, what I'm going to do is set out a case for why I think synthetic biology is going to be one of the very important tools that we're going to need going forward with respect to some of the major challenges we face over the next 35 years in terms of food production globally. So first of all, food and synthetic biology don't always sit very closely together or comfortably together. I was involved in the Research Council, Cross-Research Council dialogue with the public back about 10 odd years ago. And one of the things that came through very strongly was that people felt that food was one thing they didn't particularly want to have monkeyed around by the synthetic biologists. But I'm going to set out a case here that actually I think it's something, a very valuable tool we want to now revisit, having learned a great deal over the last decade. So 2 scenarios, nightmare future, or a really important lifeline that's going to help us address some of the very major issues we're going to face in the next 30-odd years with respect to providing a safe, nutritious, and adequate food supply for the world. And in fact, it may be one of those very key technologies that guide us through John Beddington's perfect storm to come of our depletion of resources, climate change, and population growth. And the picture I put there on the other side on the right-hand side about with the chemists, it's kind of interesting. I wonder if they had a debate back in the 19th century about the horrors or the benefits of synthetic chemistry, which were completely new at that time. And now, of course, we realise it was one of the game-changing advances that came through of the 20th century. So why do we need synthetic biology in the whole area of food production? I would argue that we need it because we're going to have to change the way we do things faster than we've ever done before. We're going to have to prepare make more than 60% more food available between now and 2050. And that means that we're going to have to feed as many people in the next 40 years as we have in the last 10,000. That's an amazing challenge for us as biologists, chemists, and agricultural scientists. In the past, we've been able to be buffered, I think, against food variabilities in supply because we've been one of the nations of the world that can just go out there and buy what we want. Increasingly, that's no longer the case. Many of the nations we're now competing with for food, which have very large populations, are able to do that just as easily as we are. And we're also facing something else for the first time in human history. There are more people who are obese in the world than are severely undernourished. So there's something going sort of sadly awry with our kind of view of where we're going with respect to food production in the next few years. Now, faced with these challenges, you'd think that agriculture would be really fired up and ready for the major challenges to come. In fact, agriculture has been systematically underfunded for the last 25 years. This is a Weekend Review article from about 7 or 8 years ago, just forecasting the end of UK farming. Why bother? Just turn the place into a golf course. And on the other side, one of the outcomes of that has been is that since 1995, we've seen some of our major crops. In this case, these are DEFRA statistics on winter wheat production. And they show that since 1995, we have stalled in the production of wheat per hectare at around 8 tonnes. And that figure has not been improved on. Lots of work has been done, lots of inputs put into agriculture, but we haven't shifted that. And that is not an unusual figure now. Many of our major commodity crops around the world are not expanding in terms of their productivity. They're either static, they're going up very slowly, or in some cases actually receding. So we're not particularly well placed to deal with some of those major challenges I had in the previous slide. Now, the good news is we've done all this before. In 1945, Norman Borlaug and others realized the world food supply was in a complete mess. And he got together with other scientists, with geneticists, with chemists, with engineers, and together they pulled off one of the greatest feats, I think, of scientific achievements of the 20th century. They enhanced food productivity, typical in cereal, staple cereals, by around 10% per annum for over 20 years, and founded the basis of what we enjoy today, which is a safe, nutritious food supply. And they did that by introducing quite scary technologies at the time, agrochemicals, which had never been used before. They were using new ways of generating genetic diversity in their crop plants and animals using mutagenesis. Again, disruptive, unfamiliar technologies, but we didn't seem to be terribly bothered about that at the time as being something we were deeply concerned about. I see 3 major challenges and opportunities for synthetic biology in agriculture, in particular, in the next few years, as being game changers. One of them is what I call getting through the genetic glass ceiling that we see in terms of global productivity of both crops and animals. So we see that kind of hitting a ceiling for wheat productivity. We're seeing it for many other crops. One of the reasons we're seeing that is there's only so much genetic variation and variability you can pull out of any genome without introducing new germplasm. And the germplasms we're working from are quite conserved, and it's quite difficult to do game-changing genetic transformations on them. Certainly the big game changers, like making plants more efficient in fixing carbon and in fixing nitrogen in the case of cereals, is something we can only do by introducing lots of different genes working together in a highly controlled and engineered manner. And ultimately, we want to be able to open up the ability to transform plants more rapidly through novel methods of recombination. Again, something that we could do through a synthetic biology type approach. We also need to produce foods which are better suited to the way that we live today. Most of our foods they're high in calories, but they're low in nutrients. So it's very difficult actually to get a nutritionally balanced diet from eating staple crops like wheat. So one of the things that we could be really thinking about here is how we could make food healthier for people who have more sedentary lifestyles. Type 2 diabetes is a massive problem for our population. we could re-engineer many of our starches and carbohydrates to make them much less glycemic, much less prone to turn into sugars, and therefore make them a lot healthier. And the thing I'm really interested in is what I call stabilizing farm production. We live in very variable times. For farming these days, you never know what the weather's going to have in store. The weather just doesn't come with rain and sun. It comes with pests and diseases, and these are changing at a rate we've never seen before, at a time when we're running out of agrochemicals, pesticides, and even genetic-type solutions to counteract them. So I see there's being a great market here and a great need for developing new synthetic biology-based tools, probably making them in fermenters to produce biologic or chemical agents which we can't produce chemically, using synthetic biologies to make diagnostic sensors and to engineer new forms of crop resistance. A very interesting piece of work was done in Japan after the tsunami, where they used genome editing to rapidly engineer rice plants to become salt tolerant, because they'd found their paddy fields were inundated with salt, and they could put that into elite rice cultivars, effectively coming up with a technology which would allow them to recommence rice production in otherwise environmentally damaged areas. So basically, you know, we're going to hear from people who know more about the technology at the levels of gene organization than I probably still do. But, you know, we can certainly do this at the microbial level, and I think we're making great advances now in even more complex genetic systems. We have a regulatory framework in place. We know very much the types of hazards we're likely to encounter with these organisms if they were to enter the environment. We have GM frameworks very much strongly in place in Europe, and these have been adopted in many other parts of the world as well. The question I think that we should be asking perhaps this evening is, do we have the will to see all of this work? And I would argue that it's something that as time goes by, you know, with the options running down, it's possibly something we should be making as a proactive rather than a reactive choice. So I'll just sum up here. We have a trilemma ahead of us: climate change, resource depletion, population growth, and we have some big asks that we can really make some rapid progress on if we're to use approaches like synthetic biology in sustainable production, enhancing resilience of our crops, and enhancing quality for modern nutritional needs. Thank you.
Okay, and you can hear me? That's fantastic. Right, so I'm Louise Horsfall from the University of Edinburgh, and I both teach and practice synthetic biology. My particular area of interest is applying synthetic biology tools to increase the sustainability of our bioprocesses and biomanufacturing. And that's because we have one planet. We have one planet from which all our resources have to come. And at the moment, we take, we make, and then we throw away. The UK is particularly bad at this. In 2012, and I don't really think anything's changed since, we were estimated to be 81% linear. We throw that much away. But synthetic biology offers new ways to move to a more circular economy. It's offering us new ways by which we can enable living systems to recycle our nutrients for us, whether they're biological or potentially technical as well. A fantastic example of this, a really current one, is plastics. So it was brought to the public consciousness just last year with Blue Planet 2, how much of a plastic problem we currently have, how much plastic there is in the ocean, and all the problems that it's currently causing. And without synthetic biology, we're looking at centuries before this plastic possibly is degraded enough so we don't see it. But then we're still going to have microplastics, and we don't know how long that's going to be around for. So just this week, there's been scientists who, using an identified enzyme that was identified from a bacterium in 2016 that was identified as being the first that could actually start eating plastic. This week, they have shown by mistake, but they have shown that you can improve the enzyme that this bacterium makes. What they were actually trying to do was work out how it evolved in the first place. And what they've shown is that bacteria have only been exposed to plastic for like the last 70 years, and they haven't fully evolved yet to be able to eat it, to be able to use and access its carbon source. But this then does throw up a problem, and there is going to be a risk. It depends how we use this, how we are going to go forward and utilize the fact we have now identified a bacterium that can eat plastic. If we use synthetic biology to its full potential, then why we wanted plastic in the first place, why this material was so fantastic and why we used it so much, it's potentially going to cause problems because we'll be able to degrade it, so it won't be this everlasting material. So there's two sides to this new discovery. But if we think about it now and think about it carefully and be proactive in the way that we deploy this new technology, this new idea, this new findings that we've come across, then there's definitely ways that we could start tackling this plastic problem that's man-made in the first place. An example of such a way would be maybe to take the gene that is able to degrade the plastic and add it to other bacteria. You know, we already use biotechnology for anaerobic digestion to get fuels and to basically decompose our waste anyway. But as I said, there has to be limits to this. We have to make sure that we don't just degrade all the plastic in the world, because that would be pretty useless. So this, I'll be honest, this isn't actually my area of research. My area of research, like I said, was about looking at making manufacturing more sustainable using synthetic biology. And one of the things that's a very, very established bioprocess is the production of whiskey. So, in fact, other spirits as well, whiskey, gin, vodka, they're all distilled in copper stills, and it's part of the process. It's vital to the process so that you get a really good spirit at the end of it. And one of the examples that I'm using bacterium for is that I'm engineering them to be able to transform the metal that's dissolved in the byproducts of the distilling industry and capture them so that we can reuse the copper. And this is an example of how we could get biology to help us recycle our technical nutrients as well as just the biological nutrients that we're kind of used to biology being able to recycle for us. And there's other ways that this could be deployed. We're trying to think of it in a responsible way. We don't just want to release bacteria that's able to capture metals. What we're trying to do or thinking about is ways that we could maybe use something like phytoremediation to concentrate metal ions. So that's when plants hyperaccumulate, so accumulate metals from the soil within the plants, and then perhaps we can put in place a biological process after that in a contained environment that uses engineered bacteria to be able to capture the metals back so that we can reuse them. And on that very pretty picture, I'm going to finish. Thank you very much.
Okay. So my name is Pablo Carbonell, and what I would like to talk in this introduction is about the role of engineering in synthetic biology. I work on synthetic biology, but the way that I arrived at working in synthetic biology, I think it was a little bit unexpected. When I was doing my PhD thesis, I was working in an electronics lab. I was designing electronic circuits, devices. I couldn't imagine that actually I was going to work on synthetic biology. Then I remember one day I was walking through the campus at New York University with my supervisor, Professor Young, and he challenged me about what I thought was going to be the most disruptive technology of this 21st century. So, since Professor Young is an expert in cybernetics and complex systems, my answer was probably going to be robotics or perhaps artificial intelligence. But his reply was that, yes, all of this was going to be part of it, but actually the breakthrough technology of the 21st century was going to be biology and biotechnology. That was a bold statement for me, because as an engineer it was difficult to think how to transfer this knowledge of engineering into biology, how to apply these concepts of circuits, devices. But now that some few years have passed after that, I work in SynBiochem, which is the Manchester Synthetic Biology Center. I apply the principles of engineering and machine learning into biology. Our mission is to produce fine chemicals by using sustainable synthetic biology techniques. We apply a principle that is called design, build, test, and learn cycle, which generally is found in manufacturing, is coming from there, and now we can apply it into biology and therefore create a new technology that is becoming biomanufacturing. Then what we do is to look for genes that have some property, are maybe found in some plant and some other organisms, and then bring them into microorganisms like bacteria or yeast, so that we can scale up the production of this pathway. We would like, in that way, to address some of the challenges of today. For instance, we would like to produce renewable chemicals, like, for instance, biodegradable plastics, or we want to make pharmaceuticals more affordable for everyone, feeding the world population. In general, our mission is to try to replace processes that now are done in the chemical industry in a non-friendly way from the point of view of the environment, something that is more sustainable, cleaner and greener. And synthetic biology is becoming more and more an important player on what is called the fourth industrial revolution, along with technologies such as automation, robotics, artificial intelligence, and big data. There are some projections that say that by 2020, the synthetic biology world market will reach 40 billion of US dollars. And this is because it's becoming a very powerful technology. I can now sit on my desk and just through some few clicks redesign an organism by introducing some pathways. Or I can even rewrite some genes from scratch, rewrite an entire genome, and then send that to synthesize, and when I get back this DNA, build the organism with these new capabilities, with these new functionalities. This is something that is really very powerful. And with all of these capabilities, what comes also is responsibility. So we have to also consider regulation about this new technology. In a letter that was addressed to the European Commission by several prominent scientists, including 16 Nobel laureates, they made the point that what we need to regulate is not actually the technology itself, but these products that now we are producing, these new organisms, because we have to look at what are the properties in terms of hazards and risks, but also from the point of view of ethical and societal consequences. Synthetic biology currently is facing what is called the Colling-Bridge dilemma, that says that when a new technology appears, is introduced, at the beginning we are not aware of all of its details, all of these consequences. But then when something unexpected arrives, generally this technology is already an important part of the society and of the economy, and therefore it's not so easy to control. How we can address this dilemma? by engaging in this type of public debates with all the stakeholders, including industry partners, scientists, and funding bodies or regulating agencies, as well as the general public and NGOs. But this has to be an informed debate. And this is our responsibility as scientists to inform about the possibilities of this technology, which are really providing now a really clear, bright future for us, because we are starting to produce new things that never have been seen before in nature. But at the same time also, what are the implications associated with this technology? And this is hopefully what we are going to do today in this debate. Thank you.
I was being plugged in. Just a word of introduction about myself. I have to apologise first of all, although I did a first degree in botany. I've never actually been in this building. Well, I have once before, so it's only as a botanist, it's only my second visit in the home botany. So, apologies for that historic mistake. Anyway, my own background is as a plant scientist who worked in industry for 10 years. At the days when there was ICI, which at the time was the biggest British company, it didn't live with the times, it didn't change with the times, and now ICI has disappeared. So that's a lesson in you have to change with times if you're working within science. One of the things I do now at university, but I spent the last 10 years also on the DEFRA committee, the AIM committee, that advises the UK government on new genetic methodologies and their applications in the environment. Not in contained environments, but in the environment at large. So we have applications all the time, either for plants, which is my speciality, or for GM medicines, new cancer therapies, some new microbial, new things that are put into the environment, including animals, and I can talk about GM insects as well, other animals if required later on. And I'm going to take just briefly some of the regulatory views, things that I've learned over the last few years. Regulation has been mentioned once or twice in a more or less positive environment. And one of the things that I suppose I'm going to say is that the take-home message is probably that the present regulatory system, for much of the science we're talking about, is dysfunctional and is not fit for purpose, because it was developed 20 years ago at a time when the technologies we're now discussing weren't even dreamt about. But anyway, to show that there was a history, just take you back. This is a historical building, and one bit of history that says there were a few bright people 50 or 60 years ago who could see what was coming. Not many of them, and they were decried in many ways at the time. But one was Ken Mather, you might know, the geneticist from Birmingham, who at a meeting in John Innes was asked to predict what the future was going to be. And he was world famous quantitative geneticist, and he said, if we could, what he would like in the future is a method where you could direct mutation in plant and animal breeding. Rather than combining together genomes together, you could take the best and selectively mutate it. As it says here, the dream of directed mutations too was very much a dream. There was no indication from genetics or anything else that anybody knew as to whether it could ever be done. Obviously, he had the dream, and it turned out that it was true, but it took 60 years to reach that time. In terms of new technologies for plants, and I won't go through the list because most people here in the audience probably know quite a lot about it. People have struggled to try to modify selectively, taking that lead from 50 years ago. They put genes in, and I don't want to talk about GM directly, although I see in the introduction, and I could have some sort of controversy about the introduction as it was put before the meeting here. But CRISPR-Cas as a means of gene editing was a form of GM, and as you'll see as I go through, that's a very sort of controversial thing to put together. But all of what I'm saying, really, how you modify genes, how you do synthetic biology, depends upon understanding gene sequences. And as we now know, every gene sequence of every major crop and every major animal, we know base by base, chemical by chemical, the DNA base is the polymer that makes up organisms and encodes organisms. We know chemically each bit of each gene of each organism across the world. And we can now, as we've heard, start to do very, very sophisticated changes in those DNA sequences, either in the sequence itself or in the other things that are added to the DNA, because genes are only active if things are added to repress their activity or taken away to express their activity. We have 20, 30, 40,000 genes, but obviously only a minority of those are switched on in any part of our body at any one time. Each gene can produce, in many cases, dozens and dozens of different proteins. So the one gene, one protein hypothesis is long gone. Your body, although it has the same number of genes obviously in every cell, the cells in your brain, each gene produces on average 30 or 40 different proteins. It's the most complex bit of your body. Each cell in your muscle may produce a very small number of proteins. So the complexity of it all depends on regulation of the genes, not just in their sequence. And that's part of this new revolution. So talking about international regulation for this technology, what is it and does it work? And I've started by saying it probably doesn't work. The reason it doesn't work is that we have such a disparate view of the technologies in terms of regulation across the globe. Everybody wants to regulate things that are safe, to make things safe. When the regulation started 30 years ago, the first genetically modified organism, companies and countries put together regulations, somewhat in the bizarre hope, looking back on it, that it would stimulate the science, that it would make life easier, that biotechnology would become more accessible. And as we know, that's not how it was used. Gene technologies, we've heard CRISPR-Cas mentioned in the introduction. This is a technology whereby you can specifically target any gene. You can modify bits of it, you can switch it on and off, you can make it work quicker or more slowly. In the US, most of these technologies, whether we like it or not, the lead internationally is often taken from the US. This was one of the examples that is always put forward to say this was one of the first gene-edited things that we eat, a foodstuff, a few years ago. So the genes of this fungus, this mushroom, were edited to stop it going brown when you stick it in the fridge and you cut it. It doesn't go brown. Lasts longer, makes it for better and more economical, less food wastage. We're all trying to waste less food. So in America now, all these technologies from 2015 onwards do not fall within any US regulatory process. And just at the end of March, 10 days ago, 2 weeks ago, there was a statement from the USDA, head of the United States Department of Agriculture, that they would not regulate genome-edited organisms because there was no genes put in, therefore they wouldn't regulate them because you were just tweaking existing genes. So in the US, these businesses are green light and they are all ahead. If you look at the editorials in the Wall Street Journal and elsewhere over the last 10 days, this is big news. It got briefly into the UK press in the Observer, but with a sort of slightly garbled interpretation portion. In the UK, we're governed, as we are now, by EU regulations written 20 years ago, which were meant to stimulate activity and did exactly the opposite because of the interaction between politics and science, which is very unhappy in most countries. So first of all, they have to find a definition of GM and GM organisms. And this is a committee decision, but they put forward these definitions. They didn't include humans because that's outside the remit, but they included other organisms where the techniques of GM had to involve some recombination, some recombinant nucleic acid. There were various things not considered the result of GM, so they were outside the scope. But most importantly then, there was another exclusion that even if you did use it, molecular biology, recombinant DNA methods, if you could say that it was a form of mutation or mutagenesis, then it was excluded, because mutation breeding was considered traditional, old-fashioned, and shouldn't be regulated. Now, of course, we have a set of definitions and a set of science advances that mean we've come into major conflict, and the conflict is spread across Europe. This is a production or a cartoon of the different countries in Europe and how often they reject scientific opinion based upon these new forms of GM organisms. At the top you see certain countries, regardless of the organism, they always vote against it in the EU system. So it's red all the way across. On the bottom you've got some countries that vote in favour of the scientific decision most of the time. And you've got a few in the middle who like it both ways, including France and Germany, who quite a lot of the time abstain, or they're not in the room when the votes are taken, so they can't be accused of voting against. So there's some really complex politics, and that's why we have this deadlock in terms of science and applications of science and the benefit of European science. And it's why we're at this point of time when we're trying to sort of plough through this morass. We've got this impasse. It's almost impossible to get a new product in the old GM fashion through this regulation because you never get a qualified vote one way or the other. It goes to the Commission, they vote one way, they make an opinion against it, and so on and so forth. So the question now, and it's a key question for the legal system that we're part of, is are these new technologies, are they or are they not within the existing definitions of mutation? And so there's a really important, any of you are lawyers, I don't know, but you probably know about this. There's a really, really crucial case going through the European Court of Justice at the moment. It was proposed by the French and some of the NGOs who wanted the European Court of Justice to deem that these new GM type of methods are not mutation, and therefore they should be regulated. Lots of people have put forward opinions, and I've helped DEFRA with their barristers who briefed on behalf of the UK government in this case to say this is a kind of an extreme version and should be rejected. The preliminary verdict has come out, and the way that the ECJ works is you have a preliminary one and then you have a final verdict. In January this year they came out with two major statements which sound okay to in detail. The first one is that these new techniques and organisms obtained by them should be exempt from the GM regulations. And they also said member states are free to adopt measures to regulate them if they see so fit. So in other words, there is no uniformity, and this is another means of paralysing scientific activity within Europe, because as sure as anything, those countries who are opposed to it will say we regulate it first and against, so we won't, and that means that the commercial investment and activity within Europe is basically declined to an even lower level, because no commercial company wants to take any of these methodologies and put them into a marketplace unless they're sure of the size of the market. And Europe as one market is okay, but Europe as 28 markets with over 28 opinions is not okay. Then you say, what about Brexit? We're going to get out of all of this. And in theory, yes, we can, but is it worth the effort? Because we can move away from the ECJs, as people want to do, we can move away from this legislation, but we're left with the UK, and we're not even left with the UK because Scotland and Wales are not in favour of this legislation. So we're left with England, and England is not a big enough market. Even if we rewrite legislation which is fit for purpose, England is not a big enough market for any company to consider to invest. And that's rather depressive prognosis, but that's sort of my verdict in this particular debate, that we're in a very, very hard place when it comes to regulation. We've got some really fantastic science, but we don't have a framework in which to use that science. So I'll stop.